Why don't we get started? Welcome to Epidemiology and Community Health Seminar. My name is Rachel Whittam, and I'm the person who usually kicks things off and introduces people. Ruby Wynn is not here today, so I'm sort of like the substitute teacher. It is really exciting to introduce Dr. Rick Jansen. He is actually someone who's rather familiar with Minnesota and the University of Minnesota, having gotten his master's and PhD here in the Division of Environmental Health Sciences in 2009. He has done, he did a postdoc in genetic epidemiology at the Mayo Clinic, and also another postdoc at the University of Chicago in molecular epidemiology. And he's currently an associate professor at North Dakota State University. But he's coming to us rather soon, or nearer to us, I should say. He's going to be joining the Masonic Cancer Center in January as a biostatistician. And he's also going to be, hopefully we anticipate he'll be an adjunct faculty member in our division as well. His research interests include, it's probably obvious, genomic epidemiology, cancer epidemiology, spatial epidemiology, epimethods, bioinformatics, and dietary intake assessment. And he's currently a part of multiple funded projects from national funders, including NIH, the Department of Defense, the National Science Foundation. So I think everything's working and everything's recording and people are online. I'm going to ask that people save questions to the end. Those of you who are online, when it's question time, we're in this like higher tech room, so you can just like use your voice to ask a question. You don't necessarily need to do anything special on Zoom. You can just unmute yourself and use your voice if you have a question. So thank you so much. Great.
Thank you, Rachel. All right, microphone's working? Great. So I wanted to thank Rachel again for the introduction. And I wanted to take this opportunity to come present some of what I do, some of my background, given that I'm coming to U of M in January as a biostatistician for the Cancer Center. So I'm framing the presentation in the sense of trying to establish some collaborations within the department. I'm excited to join U of M and in the Cancer Center, but also potentially through an adjunct position in the future within your division, the Epi and Community Health Science Division. So given that most people in the department, at least according to the website profile, do not do cancer or genomics or genetics, I'm trying to bring in some framework, how you can organize your thoughts around some of your own research interests. I'll also try to highlight some of the overlap in work I've done in my professional career that may overlap with what some of you are currently doing. And just this idea that, you know, based on background, people have different experiences, they can enrich different teams. And so when you do team science, you form a team in your projects, you're going to develop stronger research questions and potential solutions. So just keeping that in mind. All right, so again, today's purpose, introduce myself, provide this context framework that you can apply to your own research or collaborate with me in the future. We can discuss things or just informal discussions. And then demonstrate how I'm applying this context framework to pancreatic cancer. All right, so background, I got my PhD and master's here at U of M in environmental health sciences with a minor in biostats and epi. I work with Dr. Church as my main mentor. I also worked with Bruce Alexander and George Maldonado. And then my dissertation focus was on taking a look at case ascertainment bias. Whenever we are enrolling people into our studies, whether they're screen detected or not screen detected, if that's differential between different groups that we're taking a look at, that's going to influence who's in your study and affect your observed odds ratio. Then I did a postdoc at Mayo Clinic. There it was in genetic epidemiology with a focus on pancreatic cancer with Dr. Gloria Peterson. I continue to collaborate with her, and I get all my samples from her in the genomic analysis work that I do currently. And then I did I also did a lot of writing about the food frequency questionnaire data and trying to make connections to dietary components in pancreatic cancer. Then I did a second postdoc at University of Chicago. There it was more genomic epidemiology. We were looking at a Bangladesh population, so they have high arsenic exposure through their well water, and so they develop skin lesions as associated with that. We were looking at if there was a way that we could take a look at a subgroup of the population that might be at increased risk of developing skin lesions, maybe at lower doses compared to the rest of the population, using principal component analysis. We also took a look at the population to see if methylation markers were associated with age and evaluating that. So just some collaborations that I've had during my time at NDSU, making a connection and building off the foundation in my training and education, the epidemiology, biostats, environmental health, and bioinformatics, and just working on different types of cancer, breast cancer, currently collaborating with EVMS, an investigator there, and then also pancreatic cancer, continuing to collaborate with Dr. Gloria Peterson. And then it's been very interesting in my own research program to sort of get into all the visualizations that we can do, all the processing that we can do related to bioinformatics and the processing of this genomic data sets. But even beyond that, like I mentioned earlier, trying to apply what I'm talking about to the broader research environment as we get wearable technology, as we do continuous monitoring, we're going to get more and more data points. How can we organize those data points? How can we manage those data sets and get meaningful results, meaningful visualizations out of those data sets? And then in terms of environmental health, the arsenic related project at the University of Chicago, and then some pediatric related research regarding obesity, risk factors associated with obesity. Currently working with a colleague at NDSU, looking at trying to assess the impact on obesity in children during the pandemic, and to see if there is any educational components that we can implement moving forward to sort of bring down obesity rates that sort of increased during the pandemic. All right, so here's a couple word clouds that I created. The one on the left is my research interest and skills. The one on the right is the one I pulled from the faculty members that had research interests listed on the website. And so we can sort of see that with the colored version of the one on the right, we can sort of see some overlap related to pancreatic cancer in terms of some risk factors like diabetes, obesity, nutrition, and food related to the dietary assessment work that I have an interest in doing, and then the genetics and genomic components, and then of course epidemiology. So it's a little challenging because there's slightly different words, and so those get presented as different words, but the idea is there. All right, so take-home message from the presentation today is context is important. In traditional epidemiology, we talk about that in terms of stratification. So we'll talk about that as we move through the slides a little bit more. that transdisciplinary collaboration is so important, forming those teams of people with different backgrounds, different expertise, help you develop a well-rounded type of solution and approach, and you're likely to be much more successful in your research program. And then technology, again, wearable technology, we're going to get more and more data, continuous monitoring. How can we manage all that data? How can we use it? How can we effectively, so let's talk about maybe public health app. How can we give individualized type of messaging to our population as opposed to these general messages? And people say, okay, I'm supposed to eat 5 pieces of fruit a day and that's supposed to keep me healthy, but then I get this disease. So how can we potentially, or I'm supposed to lose weight if I exercise an hour, a half an hour per day, 5 times a week, but I'm not seeing the weight come off. Well, maybe there's some metabolism pieces that are at play there. And so how can we get buy-in from the population to sort of create more personalized type of messaging that may be more effective for individuals within the population? Different discussion. All right, so the larger data set necessitates the movement towards a causal type of approach as opposed to an association test. So we know, as we'll see as we move through the presentation, we have so many different genomic markers. There's so many tests going on. Statistical significance is only a piece of the story. And so how can we move to more of a biologically informed type of modeling and evaluation and move away from completely relying on the statistical piece? All right, so moving on to the context framework. So traditionally, we talk about this in terms of stratification, but we can see from the 2 tables here that number 1, it's going to be, we're presenting it as dichotomous, but it could be categorical, but not continuous. So that's a potential limitation. Another thing is that see how many boxes we have on the right diagram when we've only evaluated 3 different variables. So it becomes tedious to create all these tables. Obviously, software can do this, but you still have the limitation of not having a continuous variable and evaluating those. So you can see 30 strata, 4 cells in each table, 5 individuals. We have 270 as a minimum sample size that we would want to have to fill out these tables. And that would just be a minimum. And then we wouldn't have any associations because every box would be filled with 5 people. So, all right, so expanding beyond that, we oftentimes in Epi, a standard is using regression. So here we can evaluate continuous, we can evaluate dichotomous categorical variables. We can evaluate many at the same time. We can adjust for those in the model, but obviously there's limitations to the way that we present our variables and how effective, based on the way that we measure the variables, that adjustment is to avoid potential compounding in your study. We can also see from the scatter plot there that when we look at the lines of best fit created from the regression, that we have a lot of variation around that line that is not necessarily captured by the average. So how can we capture some of this variation, understand what this variation means? How can we maybe stratify further, understand the context as what's biologically important in these individuals' lives that is causing them to have this specific phenotype, whatever it might be, or this demographic characteristic. What's important about that related to the disease that we're trying to take a look at? All right, so presentation of trying to understand adjustment stratification in the context of genomics. So just the genome itself has over 3 billion base pairs. So those number of tests, multiple adjustment correction, trying to test for those and to get meaningful results is very difficult. So at this point, basically, when we're doing any type of genome-wide type of associations, we're just trying to do exploratory type of analyses, trying to understand what might be most important, what could potentially be picked up. So we're missing maybe some of the variation, some of the individual factors that might be important to a subset of the population that we can't necessarily capture when we're looking at the broader scope of things. And then if we throw into that not only the genetic part of things, but then if we start considering epigenetics and we look at gene expression, if we look at microRNA, non-coding RNA, if we look at methylation markers, if we start taking into account 3D structure, the organization of the genome itself, all of those factors exponentially will increase the number of tests that we need to do. And so again, trying to create the story that we cannot just rely on statistical significance, but we have to move to incorporating more biological pieces of information. Animal models, laboratory models are a great way to infuse some understanding as to what might be going on biologically and to how can we develop these workflows, bioinformatic processes and statistics to incorporate some of that laboratory type of analysis and information that we can gain. And then I also have some images here relating to different types of tissues. So some disease is more systematic than others, but as the research field moves forward and we collect more information, it's becoming clear that that disease is not going to be confined to one organ or one specific area. There's a lot of crosstalk, even within the micro or macro environment between the body and body organs. And so what's going on there? How can we capture that information? How can we use that information to make more informed decisions and more informed type of hypotheses to move forward with and do additional research on? All right, so if we add on top of that, spatial and temporal type of associations, so are we collecting data over time? How are we incorporating that into our models? Are we collecting data at different scales? So are we looking at just the United States? Are we looking at within different communities, within a specific state? And how does social structure play a role in disease development? And how can we take that aspect of things into account? So the overall context, so a wider context of not only the individual cell, the individual organ that's affected, but how does everything else that surrounds the individual impact their disease development, but potentially the progression. So if they have a stronger support system, if they have a different type of upbringing in terms of how does that prepare them to be resilient? How does that prepare them to sort of face a challenge and overcome that challenge? How is that going to influence how their body is responding to whatever disease they might be looking at? And so taking those aspects into account as well. All right, so it's clear we have a big data problem. Data sets are getting larger and larger. Sample sizes are always going to be limited. So that means statistical power is going to be limited. And oftentimes the assumptions that we're making when we're using parametric type methods like regression do not hold. So again, thinking about how we can move from an association type of a test to a causal type of framework. So how am I applying the context framework to the work I'm doing in pancreatic cancer? So again, context is important. What type of pancreatic cancer are we talking about? So it's not one disease. There's several different types of pancreatic cancer. How are we classifying those different types of pancreatic cancer? So if we just compare different types of cancer to one another, we see on the bar chart on the very left-hand side of the screen that pancreatic cancer as a whole type of disease has the worst survival of all the other common types of cancers out there, if we're just combining all the types of cancers together. And then when we subgroup pancreatic cancer into location of the cancer, we see some separation in terms of survival. We also see when we look at different types of stages, the later stage is obviously going to have worse survival, being that it's metastatic and spread throughout the body. And so we also see, depending on location, so the plot up on the top right-hand corner, we see that the rate of disease and the associated death is much different when we look at Europe and the United States, for example, compared to the bottom of the chart, looking at different regions of Africa. And so trying to understand that context. So as I mentioned, there's different types of pancreatic cancer. The most common type is adenocarcinoma, with over 90% of pancreatic cancer being diagnosed as that. When we look at survival rates based on different stages, we can see that as we move down the chart from localized to distant, the survival becomes worse. We're detecting the disease at a later state. It's more advanced, and so treatments are not as effective. And so how can we think about moving the diagnosis date earlier? But again, we have to consider lead time bias in those situations. So if we're detecting somebody earlier, are we truly improving their quality of life? Are we truly increasing their length of survival? Or are we just increasing our visibility of their observed survival? And so trying to understand that component as well to make sure that any medicines we're using are effective, actually effective. Looking at risk factors, tobacco use, obesity, diabetes, chronic pancreatitis. So most of these emphasize sort of a state of dysfunction, a state of inflammation that potentially leads to the cancer. So diabetes, it's really only associated with recent onset, so that's likely more a reflection of the presence of disease as opposed to actually causing pancreatic cancer to develop. But why it's important to think about this as a factor to look at in your studies is that you could identify a population that might be at increased risk. So if we identify those with recent diabetes, they have a higher rate of pancreatic cancer development than the general population. So can we target that group of people for some sort of early detection method or intervention? And so we can use that risk factor in that regard. So age, just a breakdown in DNA repair, general breakdown in the processes of the body overall being associated with pancreatic cancer. Genetic syndromes, gender and race, all being associated in different capacities with pancreatic cancer. And then the overall, and we can see the distribution as we look at the different plots between some of those risk factors and age. And so we can see that they have a different age distribution. And so when we're sampling the population, we have to think about this different distribution when we are enrolling individuals into our study. And if we identify individuals at an earlier age, they're likely to have a different risk factor profile than those that we enroll at a later age. So only about 35% of pancreatic cancer development is associated with currently known heritable factors. And so a large percentage is still classified as sporadic. So how can we use genomic analyses, and as the technology continues to advance, to understand what actually is associated with nature versus what might be associated with sort of their environment that they are developing, and sort of understanding how those interact with one another to spell out more of the 60% piece of things, so that we can develop public health interventions, early detection approaches to help with pancreatic cancer development, and at least improve quality of life. So generally, one of the models is that the pancreas moves from a normal state through several different panning stages, and then eventually develops metastases. With the tumor, with the microenvironment, modifications happening there, as well as modifications happening to the pancreas cells themselves. So again, we can use the animal models, the laboratory environment, to understand some of these biological changes that might be happening in order to maybe detect these changes earlier on and intervene in some capacity. So as we look at the top, there's some driver type of mutations that have been identified as early mutations that happen in pancreatic cancer related to KRAS being an oncogene, and then several different types of tumor suppressor genes like TP53 and SMAD4. Just looking at KRAS, one of the earliest type of driver mutations that we see in pancreatic cancer, we see that overall there's a difference when we look at normal tissue compared to adenocarcinoma tissue in terms of their mRNA or gene expression for KRAS. We also see that we can look at the survival curve for those with low KRAS expression versus high. Those with low KRAS expression have much better survival than those with high expression. And when we talk about the way that we sample tumor tissue, we have to take into account different devices that are potentially used. So if we're doing a needle biopsy like we do with pancreatic cancer, how likely is it that that biopsy is going to contain only tumor tissue? Not very good. So how can we adjust our analysis to take into account that it's not all tumor tissue, that there's some surrounding cells that we get in there. So when we're combining the DNA together and analyzing what expression levels there are, we can adjust out potentially the expression that we would expect to see based on cell type. And so the tumor purity is one way that individuals have tried to sort of adjust for the type of sample that was collected. So the more pure the tumor, the more the mutations, the epigenetic changes are likely cancer related as opposed to maybe cancer environment related. So piecing those two things, separating those two things from one another. And so the way that the sample is collected, how the sample is stored, how many tumor cells are within the sample itself are all important factors to take into account. And then just the Venn diagram here, making the connection between genetics, epigenetics, and environment. All of those are working together to create the phenotype that we see for an individual. So how can we develop a model? How can we develop our research to understand or account for these different components to really understand for an individual what leads them to develop disease, what leads to their prognosis once they do develop disease. So the intriguing thing of epigenetics to public health is that that's potentially a way that we can adjust or intervene on somebody's genetic material. So we think that the genetic is static, it can't really change it, but potentially with the epigenetic factors, if we can target some of these pathways, we could impact somebody's genetics. So we could overcome genetic susceptibility with some of these epigenetic factors. So that's, I guess, the main idea and purpose as to why, as a public health researcher, I want to study epigenetics. We also can potentially understand biologically important types of changes that happen with a specific mutation. So is the mutation itself causing a functional change within the cell because the gene itself is expressed differently, or is it that now it no longer is able to bind with certain proteins within the cell, and that's what is actually causing the functional change. So trying to understand that piece of disease development. And then just thinking overall, how can we move? So in epidemiology, much of the data that we collect is self-reported, right? So there's a lot of error. People recall things differently, especially when we're talking about food recall. We ask them a week ago, a month ago, a year ago, 5 years ago, what did you eat on average? It's people recall things differently. Maybe those that have cancer recall it better than those that don't because they've been thinking about it, maybe not. And so it's probably tied to education, definitely tied to career choices. And so how does that influence the results that we're obtaining? So can we move to trying to move the measurement of some of these components into genomics and epigenetics? Can we define, for example, diabetes, or can we define obesity and what it means for that individual in a genomic way, as opposed to asking them to report a laboratory value, or maybe using both together and understanding how does that improve our measurement? How does that improve our understanding? So for a bioinformatics approach, there's been several methods developed to try. Diablo is one of them, to try to connect these different genomic components together. So bring in these different data sets, so mutations, microRNA, gene expression, methylation, combine those different data sets together, do some pre-processing. It's going to be any of these models. It's always important to filter out markers that are deemed not important or correlate highly with other factors. So you don't necessarily have to look at all 12 factors. If 6 correlate with one another, you only have to use one of those factors to identify those as an important pathway or an important group. And then doing some discriminant analysis. Once that data is combined, what does the data tell us in terms of organization? How should they be grouped together? And then seeing how that plays out in terms of networks. So how do these different epigenetic and genetic components fit together in a specific network? So then we can move from the 3 billion individual base pairs to organizing the data in maybe pathways, biological functions, and trying to understand the data and the biologically important pieces of the data in that context. So just some additional visualizations in terms of separations between principal component groups that have been created. the heat map showing different expression profiles across those different principal components. And so we can look at all these things. And the last orange and red bar graph is sort of highlighting what some of the defined pathways are that have been identified based on this marker set. So like I mentioned before, pancreatic cancer is heterogeneous. So it's not one type of disease, but there's several different mutations going on within the tumor environment. So it's not only that the cells that are classified as tumor have mutations and changes, but also the environment is changing as well to support the survivability of those cancer cells. So what changes are happening to the cells themselves? What changes are happening to the environment? How does that impact for example, the treatment distribution to the tumor cells? How can we potentially understand the changes that are created? And one, if we're using nanotechnology, maybe we could potentially target those changes that separate the normal environment from the altered environment. Or two, we could think about ways that we could target epigenetics to reconfigure the local environment to make it more normal and accessible to potential drugs. And then the idea related to bulk sequencing. So there's been a movement to single sequencing. Traditionally, it's been more bulk sequencing where you just take a sample of the DNA within the complete environment and you just evaluate that. So if the mutation or the change happened early on, if we're doing the bulk sequencing, we're more likely to identify those changes that happened early on if they've progressed and populated the environment. Now, as the environmental changes are different, as the tumor environment changes, what happens to those tumor cells that have certain mutations and those that developed additional adaptations after? And so I think that's still an open question. How is the tumor environment changing over time? What is that doing to clonal populations in terms of what we can detect, what we can't detect? And at different points in the disease, we would likely see different types of epigenetic changes happening. And so how can we use animal models in the laboratory to understand what those are at different points? So that when maybe we detect an individual with cancer, we can understand sort of where they are in their progression of disease and really understand how we can target or slow down or prevent the progression of that type of disease. All right, so one way that individuals have tried to use both mutational profiles and transcriptomics or gene expression profiles to use principal components and classify different types of pancreatic cancer based on these global type of profiles. And so there's a way that they've separated these with principal components, and then they show that there's a separation or a difference in survivability, potentially a difference in response to treatment. And so just looking at the bulk sequencing data, we can subgroup the disease and potentially apply different types of treatment to impact survival. So here's some of the single cell sequencing information. The fact that we can get a picture of, so one, we can take a slice of the tumor environment. And so again, that's a bias sample. It only represents a portion of what's there, but then you can arrange the data in a way that you can identify the different cells that are within that environment. Now there's technology available where you're taking hundreds of slices across the tumor environment piecing those together so you get a better understanding of what that entire tumor and that entire environment looks like. So you can see where different types of cells are, they color code them within that environment. You have an understanding about, in the normal situation, maybe we don't see these cell types in this specific region, but now we see these cell types. So how can we use that? How can we use that information to improve treatments, improve our approach to improving quality of life for the patients? We also, so originally it was not spatially related, but again, as technology has advanced, they're now able to pinpoint where within the tumor the sample comes from and also reconstruct in a 3D environment where those cells are located. And out of that research has come that level of crosstalk between different types of cells. So pancreatic tumor cells are talking to the stromal cells, for example, and they are talking with quotes, and they are influencing some of the expression profile, some of the epigenetic pieces, the way that the stromal cells are behaving. And so they're enabling the cancer to progress beyond that specific environment. All right, so there's lots of data. How can we move towards the causal inference type of approach? So I've outlined here and thought about it in terms of the component cause model from Rothman. How can we, first of all, move back and develop those directed acyclic graphs based on biological information? So when we are creating these associations, when a arrow is actually there, it's something that means that there's an association. And what's even more important is if we erase or delete an arrow, that's telling us that we know that there's no association there. And so whenever you don't see an arrow, it is a little bit more meaningful than if you actually do see one, because you initially draw it with all the arrows and erase those that you know there is no association. The tricky thing with the DAGs and thinking about causal models is that how are you building in time components? So for example, if you look at, let's say, dietary intake, well, at the beginning of a disease, what is your dietary intake? How is that influencing inflammation in the body, let's say? And then as you get older, is that same food item causing additional inflammation? And so how are you building that in, that temporal component into your causal model? And so that's one challenging piece. And then the spatial component of things. So do we expect that the causal model is going to look the same regardless of the environment that you're in? No. So how we can build in some of those environmental components into the causal model, but then always keeping in mind to build in unknown factors, unmeasured factors into the model to account for some of that variability that we don't measure or we don't necessarily capture. So how can we develop these DAGs? How can we develop these causal models? Because likely there's going to be many sufficient causes for a specific disease. And so how can we build out these pie charts to understand for an individual what are the set of factors that were mutated or altered that led them to develop disease? We could develop another causal model where what are the factors that are leading to the progression of their disease or sort of their course of disease once they've been diagnosed. And so is there anything that we can identify across different causal models? is there anything that we can identify that may be limiting? Is there anything that we can, and is there a way that we can categorize different types of mutations across different models that sort of summarizes it? Like, let's say it's important to the development of pancreatic cancer that there be DNA repair alteration. So the specific alteration doesn't necessarily matter as long as there is a DNA repair alteration. So how can we look across those causal models, identify the mutations that happen within those DNA repair genes, and combine that in a meaningful way that makes sense and that we can potentially target more of a pathway, a downstream type of target, as opposed to those individual mutations that happen. All right, so now I'll just jump over to some of what I'm doing in the current COBRI project. So the COBRI project is available to universities that are under a certain funding threshold, and it's provided by the NIH. I also am using publicly available data from TCGA. And then as we move the project forward, we'll also use the ICGC data sets as well. So the main purpose is to try to understand what type of alterations are potentially important in pancreatic cancer. How can we group these alterations together in a meaningful way, like I was talking about, in terms of developing a specific pathway, specific functional alteration that we can link several different mutations together across different causal models. And then thinking about how to create the bioinformatics workflow to combine these different data sets together and build those causal models out of that. So the Manhattan plot, so across the x-axis, we have different chromosome numbers. Across the y-axis, we have significance level. And so I've highlighted a dashed black line and a solid black line. The solid black line is set at less than a genome-wide, accepted genome-wide p-value. So usually genome-wide is thought to be 5 × 10⁻⁸. Here I set the threshold lower at -7 as a way to, since I'm doing more of an exploratory type of analysis. So I'm not trying to identify necessarily only those markers that are significant in pancreatic cancer, but trying to make sure to capture all of the markers that are potentially important. So this is methylation markers. We see that it's generally pretty distributed across the genome in terms of significant markers that we're identifying.
Sorry, that was tumor tissue versus normal tissue? What's the comparison?
Yes, this one is tumor tissue versus normal tissue. And so one of the challenges when we're using many of these genomic type of testing is that there's very few normal tissue and a lot of tumor tissue. So how can we take that into account when we're doing those analyses? And so in general, it's not really taken into account when we're doing traditional type of regression analyses. So how is that affecting our results? I don't think we really know, but as we move through the slides, I'll show a way that when we use deep learning models, we can adjust for this imbalance. All right, so just some different plots here, a box plot showing that in general, we have more methylation in the tumor tissue compared to the normal tissue being the red. And then we also have a volcano plot showing us where the hypermethylation versus hypomethylation being green, and just seeing that in general in these cancer patients in the tumor samples that we have, we see an increase in hypermethylation compared to hypomethylation. So we can also create several different types of models, regression models, when we're taking a look at these results. And so taking into account purity and creating different models based on high pure sample, a normal sample, or a low pure sample, or just combining all of the samples together in terms of tumor, we see the significant number of significant CPGs that we identified in those different models. So if we focus right in the middle, we see that there's 31 CPGs that overlap between all of the different models. So it doesn't matter tumor purity, and we can identify these markers as being important regardless of whether we're making the comparison of a high pure tumor pure sample versus low tumor pure sample, or if we're just comparing tumor sample to a normal sample. Using those 31 CPGs, we can look at a heat map across the top. We see that we're categorizing those based on different variables that we can look at. So we can see stage, we can see sample type. Sample type is at the very top. So the little black is going to be the normal samples. The big brown is going to be the tumor samples. And right under that is tumor purity. So we have it classified as normal, low, high, and then unknown as the gray. And then below that we have stage. So we can see that we visually can see a difference in terms of expression pattern or methylation pattern when we look at tumor versus normal, and when we look at high versus low pure samples. But we don't really see a difference when we look at stage. All right, so when we look at the first two principal components, we can see a pretty good separation between the three different groups: the high pure sample, the low pure sample, and the normal tissue, with about 67% of the variance explained. And then an approach to try to group these alterations together is to use the something like an enrichment analysis, connecting markers to genes and then identifying which pathways those genes are linked to, which processes they're involved in, which molecular functions, which specific pathways, as a way to try to organize mutations that are happening that we're detecting into larger groupings. So that was methylation. This is looking at microRNA. So for microRNA, we have a lot less significant markers that are identified as being significant. Although we see that of those markers, there's several that are very significant. So the difference between methylation markers and microRNA markers is that usually microRNA markers will interact with many, many different genes, where methylation markers more affect just the local environment. So that difference is likely highlighted here. The fact that some of these mRNAs are interacting or altering expression of many, many genes. All right, the volcano plot looks different. We only see this time I've only plotted the green and the red. We see a lot more downregulation as opposed to upregulation. Here, looking at the correlation between genes across the x-axis and microRNAs across the y-axis, we can see some differences when we compare the top, which is the normal tissue, to the bottom, which is the tumor tissue, in terms of the way the microRNAs are interacting with different genes within the genome, And also we can see differences in terms of just the level of expression. So we have an attenuation in the tumor environment compared to the normal environment. All right, so this time when we're looking at mRNAs, we don't really see that good of a separation between the low and high. Slightly better for the normal, although it's so variable that it doesn't discriminate very well. So likely, it's because the mRNAs are interacting with so many genes that it's hard to get a separation there. And again, we can separate those out into cellular functions and pathways. And we notice that these are identified differently. These are different pathways, biological processes, and functions than what was identified by the molecular markers. So that's suggesting that these are likely complements to each other as opposed to sort of reproducing the same type of alteration and impact within the cell. So it's important to take into account those different types of alterations. All right, so then we started some work on trying to combine different data sets together. Here, we developed a process to take a look at copy number variation and also single nucleotide variation. How can we look at the burden within individuals of these four specific genes, like the driver genes? We just focused on the driver genes listed there initially. And so trying to understand how we can put these together, how can we classify these in a way that they're going to distinguish different groups of individuals from one another. So when we create a burden score, which is the single nucleotide variation plus the copy number variation times the copy number variation for that specific gene, we can see that across the genes that we looked at, the 6 genes, we can see a separation in terms of survival. And then when we build in the deep learning piece to it, we have some pre-processing that we do in terms of data organization, in terms of accounting data imputation. So trying to if individuals have some missing data, can we impute that data and get stronger outcomes from our analysis? Using the smoke correction corrects for imbalance in the data set. So it resamples and regenerates normal data, because then the normal, I think we only had 5 to 8 normal samples compared to over 100 tumor samples. So it increases the number of normal individuals within the data set that we can use in the analysis. And then we also have to do some filtering. So we have to reduce the number of markers that we're actually going to test before we run the deep learning model. And then the deep learning model sort of works like latent variable modeling or factor analysis, where it is using measured markers, reconfiguring them in a way, and then outputting a prediction as to whether this individual has cancer or doesn't have cancer. And through the, yeah.
I just wanted to say, I think we're at the top of the hour
Okay.
now. So I know that some people probably have 11 o'clock meetings and other people can probably stick around and keep going. But for anyone who has to take off, and I hopefully people on Zoom can hear me as well. I believe next week is the Martinson lecture, right? With Margaret Chesney? Yep. Okay. So that will certainly be exciting. And what else am I supposed to do according to Ruby? I'm supposed to then send them on their way. So anyone who has to go, I hope you have a wonderful Friday, and I am sending you on your way. Anyone who can stick around, we're still here, and I think we can stay in this room, right?
Yes.
Okay, thank you.
All right, thank you. So, yeah, so I guess just in general, saying that these discussions can continue. I come to U of M in January as a biostatistician in the Cancer Center. I hope to work with the department division as well. So looking forward to talking with everyone some more, understanding how I can be helpful to your team, or just organizing thoughts around your research and thinking about how we can build that out. So
Do you want to open it up for questions?
Yeah.
I don't know if anyone was able to
Good question.
stay on Zoom. People on Zoom.
all right, I think David Jacobs.
Okay.
Hi, Rick. That was very intense. And I'm glad that you're coming to the University of Minnesota again. What I'm not, I'm just starting to get immersed in these omics ideas. And I'm very sympathetic with your idea of sort of a full context. I mean, to me, we've always done causal thinking.
We lost our audio here for a minute. Can you start over?
Oh, I'm sorry. I guess I can't help it if you lose the audio. I just was complimenting you and saying that I'm beginning to get involved with this. But what I don't really see is how you put it all together and how you get to something actionable. You know, you've sort of you've laid out a whole lot of different things, which are really interesting, and the methods, I mean, they really make sense. I'm really in tune with the idea of causal modeling. I think we have always done causal modeling, you know, but it has become more explicit that that's our goal. So how do you bring it all together and make a bottom line? I mean, maybe the answer is just the same as it's always been. When you've got a sensible answer, that's probably what the answer is.
Yeah, right. So thank you for your question. The idea is, so this is still a work in progress. I'm trying to understand how we can combine all of this data together to get actionable results. So as we incorporate the biological aspect along with the statistical analysis, how can we piece those two different types of environments and data sets together to make more informed hypotheses that we can then test and write grant proposals about and see what is meaningful. And ultimately, the idea would be to design experiments using animal models or the laboratory to really understand biologically, can we influence the progression of disease? Can we influence the development of disease by making certain types of changes that have been identified using this genome-wide type of analyses? So even when we look at genetically modified mouse models, even though they're set up to develop tumors, not all of them develop tumors. So how can we use that information? And how can we analyze their set of alterations to understand why is this mouse and most mice developing tumors, but not all of them? Their environment should be the same in those settings. Their genetics should be the same. So what is the difference there? And that's an important piece, I think, that we have to continue to develop.
I guess that one really big thing is learning new computer languages and new techniques. So especially for those of us who are older and where that becomes a little more difficult, that's going to be an issue. But it seems as though you have to have vector or matrix oriented languages to do any of this. And there is going to be a lot of learning. And that itself is going to be pretty seductive because you can spend so much time learning how to do it. you may never get to the bottom line of what did we really find. So that's just a kind of a slightly pessimistic view of the future.
No, I would agree with you. And that's why I think that's why forming these interdisciplinary teams is so much more important than it ever has been, because there is a lot of expertise that you need in terms of computer science, and bringing students and staff members on board that have that expertise will make the whole research progress and more efficient. So one person doesn't have to learn how to interpret the data, learn how to write the computer code, learn how to process the data in the lab. There's different people that can do all of those things. And so forming these bigger teams is going to be essential.
And one last thing to just emphasize, and then I'll shut up, that you mentioned in one of your early slides, when you're doing this deep learning, and not in the sense of the specific method, but in the sense of all the details that you're into, you really have to remember the basic epidemiologic principles of bias and confounding and signal and noise and so on. It's suddenly, the cross-sectional data does not suddenly become, totally valid just because that's all that you can do.
I agree. It's just a tool, just like statistics,
Right.
just like anything. They're just a tool. We have to figure out how to best use those tools to get the answers we need. All right. Any other questions? I appreciate you guys taking the time to listen to my seminar. Sorry it went a little long, but the part of the purpose was to sort of start the conversation. We can continue to have discussions and conversation as time goes on, especially after I join U of M in January. So thank you.
Thanks a lot.
Bye-bye.
Stop our recording here.